briefly now to introduce Gene Yeo. So he really doesn't need an introduction. He's been pressing science in a number of exciting areas for the past decades, I'd say. He graduated his Ph.D. was from MIT in computational neuroscience. After that, he went on and was a junior fellow at the Salk Institute. While he was a junior fellow there, he also did his MBA at University of California at San Diego. After leaving his junior fellow position, he started as an assistant professor at UCSD and has moved quite rapidly through the ranks there, becoming an associate professor. And now he's a full professor in the Department of Cellular and Molecular Medicine. He's the co-director of Bioinformatics and Systems Biology graduate program there. He's also affiliated with the Institute for Genomic Medicine and the UCSD stem cell program. His lab broadly specializes in uncovering mechanisms and pathways associated with post-transcriptional control. He's developed and applied lots of innovative techniques, including the CLIP technologies and lots of CRISPR screens in biology. Along the way, he's co-founded a number of companies, one Eclipse and another Locana. And today, Gene is going to talk to us about systematic discovery of molecular and cellular functions of RNA binding proteins. So Gene, welcome virtually to the University of Michigan, and thank you for joining us.
Thank you, Steve, for the kind introduction. Can you hear me fine?
Yes,
thanks so much. Yeah, no, man, it would be so much more fun to be there in person, but I guess this is the new normal for all of us. And it gives me an excuse to put on a decent shirt here at home. Otherwise, we were running around dealing with our newborn twins in shorts and t-shirts most of the time. But really happy for this much needed distraction to talk about science. Okay, so let me see if I can go back to my pointer here. Okay, so just very quickly, some disclosures of financial interests. And hang on, I'm going to move the thumbnails to the bottom. 1 second here. Perfect. Okay. All right. So my lab works on RNA binding proteins. As we know, RNA binding proteins really modulate all the steps in the RNA life cycle. As soon as RNA is transcribed, RNA binding proteins bind and are attracted to the RNA and modulate everything from splicing to RNA decay, export, translation, localization, and even RNA granules, which given time I'll talk a little bit about at the end of my talk. And so we are very interested in how RBPs control gene expression. And clearly with this plethora of RNA binding proteins and of course hundreds of thousands of RNA interactions per RNA binding protein, disruption and mutations in RBPs, RBP-driven networks lead to many diseases, including cancers. We've been working recently on glioblastoma and breast cancer, as well as MDS, but also in neurodevelopmental and degenerative diseases, which we have historically spent a lot more effort in our research programs. And so what I thought I'll do today is actually present some of our basic studies on systematics and large-scale studies on RBPs. And then if given time, we'll talk a little bit about some of these aggregation-prone proteins and focusing mainly on technologies that we've been building that could be helpful for the community. Okay, so RBPs exert a lot of their effects by interacting with cis regulatory elements, and they can come through the recruitment of RBP to a sequence motif, or it could be through a RNA structure or a ternary complex, right? And many of these, this is still quite poorly understood. The functions of many RBPs are also unknown. So over the last, I don't know, almost a decade now, we've been very interested in understanding how RBPs work by elucidating their molecular targets, their binding sites, how they interact on genes in different parts of the transcript, and by doing so, gain insight into a variety of development disorders, human development, and diseases as well. We've leveraged this technology, initially developed in the Darnell lab at Rockefeller, called cross-linking an IP, which involves UV cross-linking a protein and precipitating the RBP RNA complex. And starting my own group, we've applied this to a variety of different organisms, systems, to address many different questions in initially viral host interactions, neurodegeneration, now in autism, cancer, and MDS. And so this has been a very useful and fruitful method to study these networks. However, in practice, though, the methods are actually rather challenging. Even instantiations of the methods like iClip sometimes require a multi-week protocol with high failure rates. There's a lot of PCR amplification in the library preps. So the data can sometimes be not quantitative. And initially when we started, we had evaluated dozens of RNA binding proteins, but now we would like to scale this to thousands of RNA binding proteins. And that's very difficult. So around 2012, 2013, we put together a team of individuals. Eric Van Nostrand led the subgroup that focuses on using now what we call enhanced clip. And Eric worked very closely with a graduate student, computational student, Gabe Pratt, who's now at a company called TwinScan. And we had many folks, staff scientists and computational biologists like Stephen and Brian that have collaborated with Eric and Gabe. And many have gone on to med school, graduate school, as you can see below, and to companies locally in San Diego as well. So Eric led this effort to try to enhance CLIP, and he took the protocol down from what was a 9-day protocol using radioactivity, radio labeling, to a 4-day protocol that didn't require radio labeling. He was able to work with folks to increase the ligation efficiencies in the protocol, such that you get the same information as, for example, iClip or a variant of CLIP, but with a 10 or 20-fold less sequencing required, and yet gain a much larger diversity in the library. And part of this efficiency and improvement is gained by not having PCR amplification as a problem here anymore. So for example, in previous data sets that you can download, where each dot is a data set here from a paper, previous CLIP data sets had a very high fraction of PCR duplicates due to the high requirement for PCR amplification, as the library preps were not very efficient. And so here's a measure of basically what fraction of the reads are not PCR duplicates, right? And so as you can see from our eClip libraries, the majority of data sets are uniquely mapped, highly not PCR duplicates, and are highly usable. And so with this method that was published in 2016, which is now probably has about 500 plus citations, we decided we were brave enough to launch a study with Brent Gravely and others in the community to go out there and map hundreds of RNA binding proteins and their binding sites. Eric and the team in the background continue to improve the technology in very short articles to get it out so that we can continue to enable the community. So I won't even sort of go over most of the technologies here, but we've also built computational pipelines to analyze the data. Everything's always been shared publicly, in open source format. And so today I'm going to sort of mention a few snippets that we had learned from our most recent publication led by Eric, one in Nature and one in Genome Biology. In the genome biology analyses were basically components that didn't fit in the original Nature publication. Okay, as I said, this cannot be done alone. So while my lab was involved and in charge of mapping the RBPs using enhanced CLIP to look for the in vivo RNA targets, the binding sites themselves are not that useful. You have to understand the function of these regulatory elements by evaluating the consequence of depletion, or localization of these RNA binding proteins. And so we work with Chris Birch, my PhD mentor, Brent Gravely, who's been a close friend and collaborator now for a long time at UConn, Xiangdong Fu at UCSD, who evaluated that many of these RBPs actually bound DNA as well. And Xiangdong Fu has also been a friend and collaborator for a while now. And then Eric Lucas' lab at Montreal that we met for the purposes of this collaboration, but it's now a fantastic friend and colleague. So we have accumulated a lot of data for many RBPs, many different dimensions of looking for in vitro binding sites using RBNS, knockdowns, RNA-seq, CHIP, and IF imaging. And so some of the things we learned very quickly was that RBPs have a really broad range of regional specificities. Some RBPs only bound to the coding regions, some bound primarily to intronic regions, some bound only to non-coding RNAs. And we were quite impressed by such a broad range of targeting and recruitment by RBPs to the RNA targets. We were trying to find ways to predict molecular functions faster. And it turns out from the CLIP data, one of the fastest ways that we had, the field came to was this metagene profile, right? So you can take an RBP and map the RBP to many genes, the binding sites, average every gene out into a fixed length, and then delineate enrichment at particular features of the gene. And so you can see here that UPF1 is enriched at the 3' UTR of a gene, and DDX3X it's a protein, it's a helicase that's enriched near the at the 5' UTR near the start codon. Now we can dig into this deeper and see that it's interesting that when you take 150 over RNA binding proteins, you very quickly see that the two main critical points of delineation is like the start codon and then the stop codon, right? You can see that the start codon, you have RBPs that are very enriched upstream of it, and then there are many RBPs enriched downstream of it, and then there are RBPs that are like right smack in the coding region, right? These profiles are quite distinct. You don't see that many that just cover across 5' to 3' UTRs, right? They seem to have specific domains, which makes a lot of sense, right? So if you look at even deeper, you can see that even among the ones that are enriched at the 5' UTR, there are ones which are right at the transcriptional start site and ones which are enriched near the start codon, right? And at the ones that are near the start site, you have the cat binding protein, even 3' cleavage factor CSTF, right? And then the ones which are at the start codon are the translation factors, including FMR1 and EIFs. So it's quite clear that the metagene profiles enable us to extract new functions for some of these RNA binding proteins. The other lesson that we learned that we could do with a large data set, generated with a unified and uniform pipeline and workflow and the same cell lines, are that we could take all the binding sites for each RBP in the two cell lines that we had generated data from K562 and HepG2 cells, and then simply compare them and ask, can you identify RBP complexes, right? And so first of all, from this plot, we can quickly see that there are RBPs that show you, well, there are RBPs like TIA like 1 and TIA 1, which are basically parallels in the same family. They tend to bind the same sites, right? So that makes sense. You have RBPs like Lin28s, right, where the overlap between their binding patterns in one cell line and the other is almost 100%. So there are many RBPs that bind exactly the same places in different cellular contexts. And then there are RBPs that are part of larger complexes, right? The 3' splice site machinery that bind the similar sites, and then also the 5' splice site machinery, including a few novel RBPs, and then completely novel interactors like this HMPL AGGF1 complex, which we then validated independently with IP Western blots. And so this seems to be a very useful way to identify RBP complexes from these co-association maps. One of the key challenges for us early on was that we thought that we were losing a lot of reads because of non-unique mapping to the genome. And it turns out that by developing a repeat family-wise alignment protocol in the group, we were able to then rescue sometimes 2 to 5-fold more reads. And we did this with the idea that if you found a read that mapped to different RNA families, we would discard those. But ones which mapped to multiple places in the genome but were found in the same family, we would keep and then maintain their mapping at single nucleotide level resolution. So here is our row 60, where in our initial standard pipeline, we actually lost a lot of reads because many of them would be thrown out because they're not uniquely mapping. But here you can see that actually by using this repeat family-wise mapping algorithm that Eric had built together with Brian in the group, you can actually retain many of these reads that map to the yRNAs. And so with this repeat family aware family mapping strategy, we finally understood why we were losing a lot of reads. It turns out that RBPs actually have substantial binding to multi-copy elements, and one could say, oh, that makes sense, right? You have ribosomal proteins here, which would bind to many elements across the genome, but actually many well-known RBPs also had substantial signal at highly repetitive elements. And so some of these elements are lines and alus that we had then gone into in Eric's paper here in great depth, identified elements, RBPs that could control line retrotransposition, others that bound alus specifically to control splicing. Here are other examples where we went in and looked at RBPs that were found to control 45S pre-rRNA processing or mature 18S and 28S processing as well. We could then map some of these RBPs directly onto the ribosome structure, and then show that they're actually distinct from RBPs that map to other repetitive elements. As many of these maps were generated and publicly released, we were able to share our data with groups prior to Eric's Nature paper. And here, for example, are 2 examples where we shared some of our maps and worked closely with the groups to identify different RBPs that controlled ribosomal RNA processing. And here's one example here where the NOL12 RBP binds here on this precursor. And then here's a map that shows a different RBP that you can see binds very differently from NOL12. And so maybe just to summarize this part of it, there's like way too much data to really go through. We quickly put, as soon as the data sets were QC'd, they were up publicly available for the community. We are continuing this project as a U grant called ENCORE, not ENCODE anymore, where the R is now RNA and not DNA. And you can see that there's on ENCODE project, there's a portal that you can access the data sets and so on. We've been able to expand quickly on the data sets that we generated and work with our own groups, but also many other groups to explore how they can be used to elucidate RNA editing, even in autism by Grace's group, and how some of these are involved with RNA granules that Roy Parker had downloaded and processed and identified in our group ones that control microRNAs. And to date, there have been many data sets that have been generated by hopefully using some of our data in the future as well. but this is only like 10%, right? And so this is actually, you know, a lower bound on the number of predicted RNA binding proteins that exist in human. You know, one could argue there's probably 2,500 predicted RNA binding proteins from work from many groups. And so there are quite a number of challenges that still exist. So I'll talk about one quick one and then take a pause, and then talk about how can we go faster and get function for other RBPs, the 90%. And then I'll take another pause and then take questions. And then if there's time permits, I'll showcase one of the directions we're going into in looking at how RBPs can be studied in the context of granule biology. Okay, so as we were pursuing a lot of our studies, we realized that CLIP gave you regional binding, but we couldn't exactly tell you which nucleotides were actually interacting with the protein. We could tell you which nucleotides cross-link to the protein, but that's different from which ones were interacting. And so we had a postdoc, Meredith Corley, that started my lab from Alan Ladderuck's lab. And she came here as a postdoc and has now recently left and joined Ionis Pharmaceuticals. But she was working on a project to try to understand how to integrate shape RNA structure probing with our CLIP technology. And so in a collaboration with Ryan Flynn at Howard Chang's group at Stanford, 4 or 5 data sets were generated using in vivo click shape or IC shape, where you have shape reagents treated in cells, and then you can extract typically what shape reagents do. As a backup, remind everybody, right? So shape reagents measure the reactivity of a nucleotide in RNA being accessible or not. And so if a nucleotide is base paired, say in a structure, then those nucleotides are not accessible, not reactive to the shape reagent. And so that's low. And then unpaired nucleotides, or ones which are not in a structural context, will have high reactivity. So that's the traditional use of shape. Here, what Meredith set out to do was to ask, and we realized this only after a lot of studies with using the shape reagents, that actually shape is interesting. The shape reagents probe reactivity of a base and assumes in the field that the base, it means that the base is base paired to another RNA. But it turns out that actually, if the base is hydrogen bonding with a protein, that base is also not reactive. And so she figured that you can actually look at protein interactions at individual nucleotides using shape. And the way she did this was by treating cells with the shape reagent, and then in 2 different contexts. One is using maintaining the protein, right? So the proteins are there. So that means that if a protein is bound to the RNA and nucleotide, the shape reagent is not reactive. But then we can also extract the RNA and then remove the proteins. And with the shape reagent, now it's reactive to the positions which are accessible because the proteins are not there. You can identify reactivity with mutational profiling or RT drop-offs, which is what we did here. Quantify reactivity rate at each nucleotide, and then just like normalize, right? So basically, we'll take the minus protein condition or protein absent condition, and then do a comparison and normalization to the plus protein condition. Here you see the ferritin light chain mRNA has ion response element, and these elements are structured or well described. And here you can see that actually with F shape, which is the difference between minus protein to protein, there's really high reactivity at this loop, and then this bulges, right? So these nucleotides are really reactive in the difference between minus protein and plus protein. because these are exactly the nucleotides which the IRE element is being that's interacting with the RRP proteins. And through looking at many X-ray structures, she was able to convince myself and the reviewers that really F-shape actually not only probes unpaired bases in structure, but actually identifies where protein RNA interactions are formed if there are hydrogen bonding. So with this large-scale F-shaped data, basically you have a map of protein RNA interactions at single nucleotide level based on their hydrogen bonding interactions. And so she imagined that with these patterns of shape reactivity, you can combine that with different search strategies to find new regulatory elements, right? And so as I talked about, the ferritin light chain IRE element has this unique sort of profile and this rough sequence. You can easily combine that to identify known IRE elements in these genes here, but also novel ones. And novel ones that we then validated with gel shifts, with the recombinant RRP proteins, and also that found that they were responsive transcriptionally to the iron levels in cells. And so this is a very useful way to identify new regulatory elements. However, like with many other protocols that requires just general RNA sequencing, you pick up the most abundant ones. What about combining this with CLIP? And so here she developed two variants. One is simply shape CLIP, right, combination, where it's a traditional shape protocol used, where you again treat cells with the shape reagent, but then ask for a specific RNA binding protein that you IP, what is the structure and binding sites of this of the RBP and at the same time, right? So as I said in shape, right, basically you get high reactivities for places which are unpaired or bound by protein, and so those are the high reactivity. But then you isolate this for like a specific RNA binding protein, so you don't have to sequence so much to look at the whole transcriptome, but specific ones you care about. She also developed the F-shaped ECLIP protocol, where basically she did the same thing as she did before, plus protein and minus protein with some adaptation, where we treat with proteases and add probing reagents at different parts of the workflow. But basically you get the hydrogen bonding interactions, right? By looking again at minus protein compared to plus protein, and there you can isolate this for a specific RBP. So she tested this for SLBP, which we know binds to the histone stem loops. Makes sense. And this well-defined structure in the 3' UTRs of histones, is identified and bound by this SL stem loop binding protein and in a particular complex. So as a proof of concept, here is our ENCODE data of just SLBP ECLIP, right? And so you can see that SLBP binds the 3' UTR of this histone mRNA. And then we tried with different shape reagents, DMS and NAI. NAI is a preferred choice because you have reactivity for all 4 bases, unlike DMS. And so you can see that they look the same. But then when you look at the reactivity maps, not just on the binding, right? You can see shape again tells you that the bases which are unpaired are very reactive, right? So that's makes sense. But interestingly enough, when you and this is true for the difference, right? This is true for the different shape regions, except NAI gives you a bit better because it gives you all 4 bases. But if you look at F shape, which is minus protein over protein, then you can see that there are bases here which are really the difference is really reactive, because this is the base which binds to SLBP and 3' H exo. And so this is the exact nucleotides with hydrogen bonding with proteins, right? And just to sort of remind us that for CLIPs, you can get RT mediated cross-link drop-offs, right? And that tells us that the RBP can be cross-linked to RNA And you can look at the cross-link points as a way to say that, you know, somewhere on the protein, some aromatic residue is cross-linked to RNA, but it is not really where the nucleotides are interacting with the protein, right? And with F shape, you can actually tell where these nucleotides are. And so these are the eclipse maps for SLBP in the RT drop-offs, just UV, right? And you can see that they're around, but they're not exactly, they're not where the interaction points really are. And so this was recently accepted. And so we think that this adds a whole new set of tools to elucidate RNA structure, but also protein-RNA interactions, right? We already know that shape is great for probing RNA structure across all transcripts. That's true. Eclipse does binding sites, but not structure. F shape, as she describes it, helps us identify RNA hydrogen bonds with RBPs, but alone doesn't do structure and doesn't do binding sites. We can add computational ways to find new regulatory elements. Combining shape and eclipse tells you binding sites and structure. And with F shape, you can do hydrogen bonds and binding sites. And then in some sort of combination with just one extra library prep, you can actually get all three. So we hope that this, you know, helps the field, you know, look for more interactions in RNA. Okay, so I'm going to take a quick pause here, and I think I'm quickly running out of time. So I want to pause and take some questions and then talk about our last part of the talk. Yeah.
Hey, Gene, this is Steve. Thanks for pausing. I think that was a ton of really exciting data that you presented, and I think it's good to give folks an opportunity to ask questions now.
Cool.
So there's a couple of questions coming in. I guess before the first question is asked, I wanted to quickly just make an observation and say thank you. I noticed that Nature paper you described was sent to review in 2017 and wasn't published in 2020.
Yes.
So thank you for your patience and for your dedication to open science through sharing data. I have a couple questions, but I'm going to defer to the community. And one question has come in, Martina. I don't know if this person wants to ask themselves or I can ask.
Yeah, go ahead. Shoot me the, oops.
Okay.
Shoot me the question.
I'm not sure if you can see the Q&A, so I'll just go ahead and ask it. Charles, are you there?
Yes, hi, I can ask it. A fantastic word, Gene. I was wondering, in your shape analysis, how can you distinguish between changes due to conformational changes of the RNA upon binding to the proteins versus direct hydrogen bonding from the protein residues?
That's a great question. And we got that as a reviewer question too. I think it's a hard one, right? Because there's no method, like it's one of these questions where if you remove the proteins, you know, in vitro, even though you folded the RNAs accurately, this is, you know, met by many of the RNA structure labs, right, as a problem, right? How do you then know that the protein removal doesn't change the structure and then affect your probing? We don't. Right. We had to keep that assumption because that was the only, there's no other way to do that that captures both at the same time. So far, at least for the RBPs and structures we've looked at, there doesn't seem to be a lot of difference by using the protease. When we went back and validate, of course, we had to go back and validate the protein RNA interactions, right, using gel shifts. But that is an ongoing assumption. That's exactly correct. Like, you know, we haven't found a way. I think the field hasn't found a way to probe both at the same time. Yeah.
Thank you.
A great question.
Thanks, Charles. Layla, can you unmute yourself and ask your question now?
Hey, Steve, I'm giving people access, but I know Peter Todd has a question. So while I give them access, let's let Peter ask.
Okay, Peter.
Gene, that was great. Good to talk to you. So quick question about, it sort of comes up from the repetitive aspect of what you were talking about. Have you looked at whether there's selective binding of multiple RNA binding proteins that simple tandem repeats? And have you been able to deconvolute those from your sequences?
That's a good question. So from Peter, right? So are there good to see you? Are there RBPs that bind to short tandem repeats? Yes. So we can actually identify them. And it's funny, even canonical RBPs that we've worked on for a long time, like RBFox2, there are actually places in the transcriptome where it's just many of these UGC sites, right? And right, like sort of tandem repeats. So even those we have found. And so yes, that's just yes. Unless you meant your question, are there like families of repetitive RNAs which are bound, like alus, antisense alus, and so on? So those we have also identified new RBPs to them. Harder to understand what they might do, but yeah, I hope I answered your question accurately.
Yeah, just sort of I mean, I think there's a lot of, as you've gotten into, and this alignment issue is a huge thing within genomic space. And I think that if you can get to some deconvolution of these repetitive elements, then I mean, maybe it's a functional component of why repetitive elements are there, because they might act as sinks or modifiers or buffers or all sorts of things.
Yeah. And now we do think that a lot of these repetitive elements do function to regulate gene expression. We haven't found though, like what we expected to find, that there are some RNAs which absorb a whole bunch of RBPs. I think the closest we came to is validating stuff like the familial binding to NORAD, right? We find that I think 30% of like palmicites are on the NORAD RNA. I think the non-coding RNA. But other than validating that, we didn't find many other examples like that. Yeah.
Great. Thanks.
Thanks, Peter.
Thanks.
I think Layla's question was that I think the grouping of RBP to localization is very interesting. What do you think the mechanism might be? Yeah, I think it falls down to their RBPs are involved in initiating translation. They tend to be at the start codon. There are ones which are involved with RNA stability, and so they're found in 3' UTR, and I guess they don't get bumped off by ribosomes. And then ones which are involved with translation elongation tend to be in the CDS. So that's how we are using these profiles to parse out potential functions of these RBPs. Yeah.
Gene, before you move on, I had a quick follow-up on that. Thanks, Layla, for the question. So all of these functions that they're characterizing seem to be post-transcriptional. You even mentioned ribosomal RNA profiles, and microRNA profiles. Have you seen any profiles on enhancer RNAs that might hint at some sort of transcriptional regulatory process instead of post-transcriptional?
Yeah, so actually there's a couple of collaborations we have with groups to look at RBP binding to eRNAs. It's just, and these other groups have been focused on eRNA, so they are trying to validate these. They found, I have a colleague at UCSD, Shannon Lauberth, that's found some exciting, evidence that some of these RBPs and eRNAs control their expression, and maybe through that control gene expression. They're much harder to validate because of the abundance of the eRNAs, but I think that stuff will be coming out over the next couple of years. Yeah.
Okay, cool, thanks. And then one more question, I think from Pedro. There was a question answered in the Q&A from Gina, so thanks, Gene. Pedro, one more question, and then Gene, I think you should move on to your second part of your talk.
Yeah, so we are looking at using hydroxy radical probing. That's something else that we're looking into. We don't know how it correlates that yet, but thanks, Pedro, for the question. Okay, so I'm going to jump back in, and if we can do that, cool. Okay, all right. So the second part of the talk, and I'll go a little bit faster here, is to how are we going to go about trying to assign molecular functions to these RBPs without 5 labs and like a lot of time and resources. So majority of RBPs still have no annotated function despite all our efforts. And so we started getting impatient, and we said, well, maybe a simple thing to do is to go back to old school methods like tethering, right? And so over the last 5 to 8 years now, we have accumulated many RNA binding protein open reading frames. We have used gateway cloning to fuse them into these plasmids and have developed a whole panel of reporter systems. And I'm gonna illustrate this by only talking about one of these reporters, right? And so this is the reporters. Oops, let me see if I can go back to the laser part. Reporter systems that have MS2 RNA hairpins in the 3' UTR downstream of Firefly. And then we can obviously swap this around and use Renilla upstream instead as a replicated study. And so the way this works is that the RBP fused to the MS2 coat protein in our constructs can be tethered by these MS2 RNA aptamers to downstream of Firefly or close to the RNA, right? And affect stability or translation. And so we just had this idea that what if we just did these hundreds of RBPs, transfected the RBP with this reporter plasmids and asked what happens to the reporter if this RBP is closed? Can that give us insight into the actual endogenous function of these RBPs in molecular assignment, right? And then if that's true, can we develop a whole range of other tethered function assays? And so we did this for this 3' UTR one to look at RNA stability and translation using initially luciferase as a readout because that was scalable. So there were many open reading frames, triplicates, transfected with the reporter compared to Firefly, and then we flipped it. Turns out the reporters have different sensitivity to being regulated, but we conservatively took the overlap for now. We looked at the validation rate by both QRT-PCR and luciferase, and that seemed to validate well. And we were happy to see many known proteins showed up to affect the luciferase output, right? So proteins that are involved with deadenylation, modification, stability, translation, capping, decapping. So those showed up, which made a lot of sense. And many of them, at least for the 50, clearly reacted the same way as if you did Firefly and Renilla. And actually, a lot of this is thresholding. I think the Renilla is a bit more noisy, but otherwise you pick up many of the same proteins. Okay. So we also validated the RNA level, as I pointed out for many candidates, whether they affected the RNA level of the reporter. And so for the most part, that compared very well with the luciferase readout. And so this gave us a way to identify very quickly. I mean, this the actual library construction took months, but the actual tethering function assay screen probably took at most a week or so. And it gives us a very quick way to prioritize proteins that may have an effect on something. And so in this case, it was RNA stability and or translation. But then we wanted to know if they actually did these things, on endogenous substrates, and we were quite surprised to find a few things. So, for example, this part is actually now known, but what we were surprised about is that the endogenous bias actually the 5' UTR. So it's CLIC3, right? And so it's interesting. You have this protein which is tethered at 3' UTR, but endogenously has the same effect on hundreds of transcripts, but actually the main bindings at 5' UTR. And obviously, we also had ones which bound on 3' UTR, which makes sense for us. So it's interesting that we actually picked up. We actually think that maybe it's because a lot of these RNAs are at the 5' end and 3' end are actually pretty close, and by just tethering the RNA close by, you are recruiting the RNP complex to the reporter. And so it's still important to do the endogenous clips to look at actually what would it normally do on normal transcripts. We were actually interested in the ones which bound to the coding regions, right? Because what are they doing in the coding regions? And how does, again, 3' UTR tethering give us this readout of endogenous CDS coding? And so UBEP2R is an illustrative example. It's a good example to illustrate. So UBEP2R binding sites by eClip is all over the exons, right? Open reading frame. So what is it doing? So from our luciferase reporters, UBEP2R is a positive enhancer of translation or RNA stability. And so we depleted UBEP2R and did RNA-seq first, and we showed that actually the RNAs most sensitive to the loss of UBEP2R were the most enriched by 8, 16, or 16-fold for being a target by CLIP studies, right, for UBEP2R. And also looking at just general translation, we find that if you deplete UBEP2R, you indeed lose protein synthesis in cells using the sunset assay. We did polysome profiling and showed using CRISPR knockout lines where UBEP2L is genetically depleted, gone, that you have a reduction of polysome association of transcripts by using really old school polysome profiling assays, followed by RNA-seq. And then interestingly enough, we said, well, this is maybe true globally. Maybe UBEP2L is somehow being, it's somehow involved with RNA stability and translation by being recruited to this mRNA. So can we actually show this? So we fused UBEP2L to this was a couple years ago now. So we did RNA targeting Cas9, where we had a YFP mRNA that was designed so that you can have multiple different guide RNAs recruit the CRISPR protein with fused UBEP2L to the RNA compared to just the Cas protein itself. And in several early papers, we have shown that Cas9, and now Jennifer's lab, other labs have shown that Cas, orthologs of Cas can also bind RNA. And Jennifer was the first lab to show that it can bind in vitro also. So we were pleased to see that actually this binding can actually recruit different proteins, and cool that we can actually enhance translation by recruiting UBEP2L to the open reading frame compared to the 5' UTR and a little bit of 3' UTR still, a little bit near the stop codon. So recruitment can programmatically enhance translation a little bit. And so how does this work normally? And so again, using our repeat element aware family wise mapping strategy, we find that actually most of the reads are not on the coding unique mRNAs, right? It's on ribosomal RNAs, and that can be background. And so we compared this to our 446 other data sets now in ENCODE for the ribosome RNA association, and you can see that UBEP2L is very much more informative and enriched at the expansion segments on the ribosome, which is very interesting. So we think what's happening is that UBEP2L actually binds and interacts with the 60S on the ribosome expansion segments. These are segments that Maria Barnard has done some beautiful work in studying that they may have cell type specific functions potentially. And so we think that this UBEP2L is somehow interacting with the ribosome, and therefore when we clip it, we clip it on mRNAs because during elongation it's coding the mRNA as well. And so our model here is that UBEP2L is playing some role in enhancing translation, and that still has to be worked out mechanistically how it's doing that, but that explains its binding across open reading frame, and why when we tether it, we probably are recruiting and pulling a local density of ribosomes onto a given mRNA target or reporter. Okay, and so that was recently also published maybe a month or so ago, which presents at this point I think the largest resource of open reading frame for RBPs, and we have both the MS2 code protein fused to it, also V5, which I think enables the community to do their own tethering screens for lots of different assays. And we have in our own lab many projects ongoing. So this was led by Enching Luo, is a grad student in my group who recently left. Also decided to go to Ionis. I think they pay really well there. And she's got many exciting RNA related projects at Ionis. And Jason Nathanson actually started the resource gathering and tethering project in my group a long time ago. He's now at the Salk Institute. Okay, I think I've sort of run out of time, so I'm going to sort of skip the last part of the talk, if that's okay, and go to acknowledgements, if I can find that slide, and just sort of end there. And these are the folks that have been amazing contributors to science in the group. And with that, I'll take questions on the second part. And thank you for your time. Most appreciated.
Thanks a lot, Gene. That's really fantastic work. And thanks also for leaving time for questions. I think I'll give it a minute for questions to come in, but before they do, I'll ask. I have a couple of things I was wondering about. One of them related to the first half of your talk, but near the end of that, where you were showing how you combine shape analyses with clip analyses. And I'm wondering if you have looked close enough at the DNA sequence to recognize any signatures of selection in coding regions that might be mediated by RNA binding proteins and not necessarily selection that's acting on the protein sequence itself. And therefore, you might be able to predict functions of human genetic variations in those coding regions.
That's a great question. we are doing those studies now. Part of the issue is with the shape studies, as we currently did it, we didn't have enough read depth to explore overlap a lot with all the the exact and sort of clean var data sets out there for variation. But we are sequencing deeper. This is part of the problem with like shape analysis, right? We had to really do a lot of sequencing to get signal at lowly, like mid to lowly abundant genes. And so we have a different strategy here. What we may do is actually combine shape with a few RBPs that we know bound to sufficient transcripts that cover what we want to look at. So that's what we're gonna do next. Yeah.
Yeah, cool. That sounds
great question. On point. Yeah.
Okay. I don't see. Are there any panelists that want to ask questions? While we're waiting for those to come in, I had another question about the second half of the material that you presented, where you were showing, I forget the name of the protein, but it's an RNA binding protein that interacts with a ribosomal RNA in a specific fashion. And you said you think it has cell type specific functions. So I guess I don't follow how that would work. How would you have a cell specific function if you're just binding to a ribosomal RNA, unless you're kind of just co-opting the transcriptome that is specific to that cell type already?
Good question. Yeah, I may have been too quick there. So Maria Barnard has been studying this different ribosomes for cell type specificity, and so that's what I meant, is that there are these expansion segments on the ribosome which she has elucidated that they might have cell type specific function because they might bind RBPs. They might bind cell type specific RBPs in that sense. We don't know yet if we have cell type specific function for UBEP2L, because UBEP2L is pretty ubiquitous. Now the expansion segments that we see that they bind to in the ribosome may be bound by other RBPs to prevent or compete for it. So we have another work in progress elucidating that for mental retardation, to look at neural specificity of loss of UBEP2L. So it's interesting. A lot of these proteins are ubiquitously expressed, but they may have cell type specificity due to its competition with other proteins that are more cell type enriched. Yeah.
Okay, I see. Thanks for walking me through that. And I can see in the panelist chat there, Stephanie Moon is trying to ask a question. I think, Stephanie, are you unmuted now?
Yes.
Great.
Awesome, thank you. So I was curious when I saw your UBOP2L data about its role in translation elongation, if there are any thoughts about how that function could be related to its role in stress granule formation, since we now know it's essential.
Yes, great question. Yeah. So we had published actually in 2018 in Sebastian's paper, Mark Miller's paper, that we had discovered that UBEP2L seemed to be important in RNA granule, stress granule biology. So it's funny, like part of my lab was studying UBEP2L and its roles in stress granules. We had depleted UBEP2L and showed that we get disease ameliorating effect in RNA like granule related ALS toxicity in fly models with a collaborator. So that was actually the last part of my talk. I didn't have time to go over, And then at the other part of my lab was looking at UBEP2L. It just so happened that they came down with this. And so we looked at this very carefully, right? And we realized that in our experiments where we overexpressed UBEP2L or depleted it, we didn't see we didn't try to we didn't cause, like, by overexpression more stress granules. So that was clear. And so we said, it probably has roles like other proteins in stress granules, found in stress granules, to have, their own roles in translation, but could also have roles in like to be recruited to stress granules, and may have roles in forming stress granules, right? It may be that it's doing, so we haven't quite deconvoluted that yet. We think that it's separable at this point, that the role in translation, recruitment, enhancement doesn't have anything to do with stress granule biology as of now, right? But however, when we knock it out, we do see initial, like, defects we do see by polysome profiling, lower translation on many substrates. But of course, when you now stress the cells with sodium arsenite, we don't see stress granules, right? Or we see a reduction in stress granules. So we do think it's separable, right? Most of our experiments are done. All experiments are done in Arjun's paper in the absence of a stressor. But then, of course, when you add stressors now, then you have this other phase of UBEP2L biology. So yeah, I'd like to hear folks' thoughts on that, actually, also, because it's been an interesting observation that we have found from these two parallel pieces of work. Yeah.
Cool, thank you. I think that makes a lot of sense. It has two roles.
Yeah, thank you, Steph. Yeah.
Okay, I don't think I see any other questions. And we're right at 5 o'clock, so perfect timing. Thanks a lot, Gene. Number one, thank you so much for all the exciting science. That was really amazing to hear from all the cool projects, and it sounds like you have 100 things kind of upcoming too, so we'll be on the lookout for that. And you also told us there's a couple new additions to your family. Congratulations again on that. Take care and stay safe, and I hope we can see you in person soon.
Thank you so much. Thank you so much, Steve.